For a bit of background, my lab is interested in head and neck cancer, where about 70% of patients come into the hospital at a late stage of the disease. Now, when this happens, doctors usually use multiple approaches in order to treat this. And the gold standard is a combination of both high-dose radiation as well as high-dose chemotherapy. Now, hearing that they're both high doses, you're probably not surprised when I say that about 40% of patients experience extremely significant toxicities. So, in order to solve this toxicity problem, our lab actually proposes to design something similar to the COVID-19 vaccine. However, instead of using lipids, we're actually proposing to use proteins as our nanoparticle carrier. And instead of loading it with the mRNA, we're loading it with that chemotherapy of interest, as well as gold, which can enhance the radiation within the tumor. Now, in order to create the system, we had to look at three main things. The first was that we needed to be able to create that protein nanoparticle consistently. And so we saw in the literature that a lot of labs were kind of just combining all the components into a glass jar, shaking it, and hoping they would get the same result each time. However, we found this wasn't the case at all, so we decided to move towards microfluidics. Microfluidics is very similar to a computer chip. However, instead of electricity running across the chip, it's actually the different liquid components that come together in order to form our nanoparticle. And as you can see in the bottom right panel in AIM-1, we were able to get very consistent results in our sizes from batch to batch. Now that we were able to create the system consistently, we had to show that we were able to get the gold inside of our nanoparticle. So in A and B under AIM-2, which is our empty carrier, you see that there's no gold present. However, D, E, and F show in red that the gold was actually encapsulated into that protein nanoparticle. Lastly, we actually had to show that our system was able to interact with X-rays and cause triggered release. So when X-rays interact with matter, they create these really reactive chemical species called reactive oxygen species. Now, these reactive oxygen species can interact with our protein and cause three main things that we can measure downstream, specifically aggregation of the protein, fragmentation, and a chemical modification of the protein. And we actually saw all three of these happening at the same time. So while we're extremely excited from the results, we still have a long way from clinical trials. So for our next steps, we first have to load the drug of interest within our system. Next, we'd like to try these with cells. And then lastly, we'd like to use this in a mouse model before moving on to clinical trials. Thank you so much for your time, and I look forward to your questions.